Assalamualaikum warahmatullahi wabarakatuh. Very good afternoon to all the participants. Um, Thank you for inviting me to this very interesting conference, and I would like to share my experience and some of our findings from the Malaysian cohort as a resource for the precision medicine initiative. As we know, there are many large cohort studies and biobanks, which are really large population-based studies. Some of them are shown here, both in highly developed countries as well as low middle income countries. And for cohort itself, as it is always linked to a large collection of biospecimen and the setting up of biobanks. There are a lot of applications or relevance to the cohort itself, or also the biobanking initiative itself. This includes being an enabler for precision medicine, and having a large data bank and biobank would mean we'll be able to do large-scale research in diagnostic and therapeutics, big data analytics, biomarker discovery and translational research, identification of risk factors, including for non-communicable diseases. It would, of course, transform healthcare as we embark on the precision medicine initiative. And of course, the adoption of artificial intelligence-driven processes and new innovation. Just an example of how large-scale studies on population samples and data can result in new findings. This was a GWAS study done on the UK Biobank to look for a new variant that could predict for the risk of coronary artery disease. This involves almost 120,000 people, and they discovered 15 new loci that increase the risk for coronary artery disease. To do such a study in Malaysia would have cost us about 36 million ringgit. This study was published in 2017. Let's go a bit into precision medicine. We probably are familiar now that precision medicine is healthcare tailored to an individual or a group of individuals based on their unique genetic makeup, environment, and lifestyle. So the aim is actually to give the right treatment to the right patient at the right time. And precision medicine tailors treatments to groups of individuals. If conventionally we give the same treatment to patients with the same disease, with the approach to precision medicine, after studies or research into large group of patients, we could cluster, or we could group patients into different groups according to either phenotypes or genotypes, and give the correct treatment or the best treatment to get the most effective outcome. So the aims of precision medicine, as I've mentioned a bit just now, is not only to give an opportunity for earlier diagnosis, more effective treatment, better quality treatment, but also to affect a large-scale effort to prevent diseases and provide more value-based healthcare and better patient safety. I'd like to give an example of two big initiatives of precision medicine. One is the famous one, the 100,000 Genomes England, that started in 2012, and they focus on cancers, rare diseases, and pathogens. They were very successful in doing this, and will target to sequence 5 million individuals. And in 2015, President Barack Obama announced the Precision Medicine Initiative in the USA. One of the big program is the setting up of a new cohort, now called All of Us, which would involve 1 million individuals who will give consent to have their genome sequenced and also for their health data to be shared. The main focus will be on non-communicable diseases. I'd like to go into an example how we can group patients according to some clinical phenotypes. This is for type 2 diabetes. This was published in 2016, where you can cluster or divide this patient into groups based on their BMI. As you look at this slide, each particular group of patients would receive a different treatment. And this is a chance to follow this kind of cohort and to see the eventual outcome of the glycemic control, as well as complications that may arise. This is another example of grouping patients based on the clinical phenotype. This is again type 2 diabetes patients. This is from a German-based study, has been published in Lancet in 2019. 5 clinical phenotypes here, including the mild age-related diabetes to mild obesity-related, those with autoimmune diabetes, severe insulin resistant, and severe insulin deficient diabetes. What's good about this kind of grouping is that, again, you can study in terms of the effectiveness of treatment, as well as complications that will arise from each particular group. So extending the discussion on this is the application of precision medicine to type 2 diabetes. You could predict individuals with a high risk of getting type 2 diabetes. You can predict response of certain drugs for treatment. You can identify those at risk of developing complication of diabetes. And you can identify classes of group of patients with similar phenotypes or genotypes. The examples that I gave in the previous 2 slides have not taken into account their genotyping status. Okay, let's move into the Malaysian cohort project. This was set up in the year 2006. And we are now a member of the Asia Cohort Consortium. And 5 years ago, we became a member of the International Health 100K Cohort Consortium, of which the membership will require to have at least 100,000 participants. So I've shown this slide before in some of my lectures. This is a GIS mapping of all the 106,000 participants based on their geographic location. And you can see the potential and beauty of doing this in Malaysia, because we are a multi-ethnic countries, and the mapping of the participant will allow a lot of environmental-based studies as well as gene-environment interaction studies. We measure the usual sort of biophysical parameters, but have some unique measurements that we have added, including cardio-ankle vascular index to look at vascular tension and vascular age. And recently, we have added retinal scan, DEXA scan to look at adiposity, as well as echocardiogram screening in a sub-cohort of individuals. Blood and urine tests, the usual fasting sugar, fasting lipids, renal profile, and full blood count, hemoglobin A1C, and soon we'll be measuring insulin levels, especially for our diabetes studies. Our lab is accredited, and we repeat this test on our participants during follow-up, that is between 4 to 6 years. Our biobank is largest in Southeast Asia. We have a main biobank and 2 satellite biobanks as backups. Some of the results, this has been published in the International Journal of Epidemiology in 2015. Most likely more female participants in our cohort. By age, this is the distribution in 2012, so it's been 8 years now. As you can see, this group would have aged by an additional almost nine years. So it's a nice distribution there, and of course this will give an opportunity to study aging as well. So this is the prevalence data. As we can see here, our type 2 diabetes prevalence is 16.6%, and we had detected about 5% of new cases, a very high prevalence of hypercholesterolemia, and 58% never knew they had high cholesterol. The same goes for hypertension, 46.5%, where 22.4% of them never knew they had hypertension. This is the overall mortality over the past 13 years since we started the cohort. 7,575 deaths from all causes and 1,506 deaths due to cancers. And this gives a big opportunity to analyze pre-diagnostic samples taken at recruitment because they were considered healthy and without symptoms of cancers or other non-communicable diseases when they were recruited. And the analysis of this, either looking at genome-based markers, protein-based markers, antibody-based markers, are ongoing. So I'd like to now share some of the snapshots of the research projects that we've done recently relevant to precision medicine. So the first one was in 2015. This was a GWAS for type 2 diabetes involving 6,000 cohort participants. We relied upon the opportunity based on the differential prevalence of type 2 diabetes between the three ethnic groups. And we recruited 3,000 with diabetes and 3,000 with normal blood sugar. So what is important here is that we identified variants for type 2 diabetes in our population. Some of these appear in the Caucasian population as well. Some of them have smaller effect size compared to other populations with an odds ratio of 1.2 to 1.4. But this is actually an opportunity for risk identification and stratification. And if you look at some of the genes that have been marked from our GWAS study, the variants of FTO and MC4R genes, as we know, causes 4 times increased risk to obesity. Screening for these variants, of course, gives us an opportunity for lifestyle intervention and also dietary modification for those at risk of type 2 diabetes. The next study I want to share is on familial hypercholesterolemia. As we know, FH will confer a risk of early coronary artery disease. It is one cause of sudden cardiac arrest, and it is actually quite common, 1 in 300. And in this study, we use the extreme value design, those with very high cholesterol levels versus those with low cholesterol levels to increase the chances of us to find new variants. So a very busy slide, but sufficient for me to say that we discovered many new novel causative variants in the first tier genes. As we know, FH is very much related to the LDL receptor genes, ApoB and PCSK9. We also found causative variants in the second tier and third tier genes. These results will soon be published. And of course, this will give us a chance to screen populations and patients who have this variant so that again, they can be treated. Hence, we can prevent them from developing sudden or other coronary artery disease. And another important finding is protective variants, which means those who have this variant will be protective for them to or against developing high LDL levels, and would possibly prevent them or lower the risk of them developing early coronary artery disease. This is the third study I would like to share. This is our study involving 36 cases who have died or individuals who have passed on from sudden cardiac arrest. We use a gene panel that covers 174 genes to do a post-mortem molecular analysis on them. And as you can see from this slide, 12 out of them we identified variants that have given them or have conferred the risk of them to develop the cardiac arrest. So an example is this 46-year-old male who were recruited at healthy at 46 years of age, but died from sudden cardiac arrest at 47 years of age. Our panel identified mutation in the KC and Q1 gene that is sort of attributed to this long QT syndrome. So these patients are at risk to have a sudden arrhythmia, and if we had, if he had been identified with this particular mutation early on, he could have been treated with beta blockers and anti-arrhythmic agents. The next study is looking at a sort of markers that could predict diabetic kidney disease, because we followed our participants over time. We could identify those with diabetes and look at their kidney status, and over the years to look at the progression of the diabetic kidney disease. So for this study, we identified one genetic variant, the KCNQ1, again familiar from the sudden cardiac death, and 3 environmental factors. So what we have done is use genetic information, lifestyle, and environment to help cluster or identify group of patients or group of participants with type 2 diabetes who are at a higher risk of developing or progressing in terms of diabetic kidney disease. So this is a new project, an ongoing project funded by MRC UK. This is a precision medicine research project for type 2 diabetes, where we will be recruiting 5,000 type 2 diabetes patients, doing genetic analysis using genotyping, doing retinal scan, to look at vessels and retina, echocardiogram, and renal function. And these are some of the ongoing projects that are related to precision medicine as well as biomarker discovery. I just go through this. Adiposity, this is our collaboration with Oxford. I mentioned precision medicine for diabetes, non-coding RNAs in diabetic nephropathy. This is going to be completed soon. Looking at biomarkers for non-alcoholic fatty liver, diabetics have a high risk of developing non-alcoholic fatty liver disease. Of course, as I mentioned, the cancer biomarker discovery project using the omics science, proteomics, and metabolomics, and so on. And intervention study on pre-diabetics, which we have planned quite some time. This is the next one is looking at mental health and cognitive function. So this is again a strategic project as our participants becoming, an aging group as well. Biomarker-based nutritional studies, and of course, gene environment studies. There are also more potential projects for precision medicine. We are planning to genotype all 120,000 participants with a funding sort of mechanism from an international partner. Biomarkers for early dementia, I mentioned this earlier. Biomarkers for early detection of cancers. And of course, in the current COVID-19 pandemic, this is an opportunity to look at genetic risk factors for COVID-19 infection. So this is this slide shows some of the challenges that we face when running this kind of large population studies in Malaysia. Health events, these are mainly by self-reporting. Of course, we try to make sure it is as accurate as possible through sort of a very detailed interview, as well as asking participants about the exact medications they are having, and also showing us whatever medical notes or in their sort of patient follow up card and booklet if they may bring with them. It's difficult to link to medical records because our participants are recruited from all over Malaysia, basically to validate events. Hence, again, the coverage for electronic medical records is still at a low stage in Malaysia. 40% of the deaths are not certified by medical personnel, meaning they died at home. Hence, we will be embarking on a verbal autopsy project to sort of help validate this mortality causes. The follow-up rate at the moment is 60%. We've entered our third follow-up stage. The attrition are mainly due to change of addresses, change of contact numbers, and so on. So in summary, I hope you would have been convinced that the Malaysian cohort is a valuable resource for research in non-communicable diseases, and I believe it would be a key enabler for precision medicine, similar to the UK Biobank, the Precision Medicine Initiative in the US, the All of Us cohort, as well as the 100,000 Genomes England project. The participants are well characterized and with data on key exposures, which is a multi-ethnic cohort, and also consists of urban and rural participants. The biospecimens of high quality. We believe the value will increase with time. We have key research projects with special interest groups. We have collaboration with other cohorts and biobanks, and of course, we are participating in global consortiums. I'd like to acknowledge the contribution of our funders and the Ministry of Science, Technology and Innovation, Ministry of Higher Education, of course, the management of UKM, UMBI, all the staff members and management, all my collaborators and co-researchers, and last but not least, the Malaysian cohort staff. Thank you very much.